So today I'm going to talk about Agrobacterium Tumefaciens by R. Yogesh, first year MSc Biotechnology. What is Agrobacterium Tumefaciens? Agrobacterium tumefaciens is a gram-negative bacterium which is the causative agent of crown gall disease in many dicotyledonous plant species. The disease symptoms results from transfer, integration, and expression of specific DNA fragment, also known as tDNA or transferred DNA from the bacterial tumor-inducing plasmid called Ta plasmid into the plant cell genome. The machinery needed for generation of transferred tDNA and for its transport into host cell is encoded by series of chromosomal and Ta plasmid encoded virulent genes. Interestingly, although the bacterial proteins possess many of the functions needed for transformation process, host plants components are also essential for import and integration of tDNA into nucleus. As I already mentioned that Agrobacterium is a gram-negative rod-shaped bacteria which has the natural ability to transfer DNA into chromosome of the host plants. This bacterium, which causes crown gall tumor in plants, contains a plasmid called or also known as Ta plasmid, which is a large circular double-stranded DNA that can replicate independent of Agrobacterium tumefaciens genome. The ability of Agrobacterium tumefaciens to induce crown gall disease in plants is controlled by genetic information carried on Ta plasmid. It has two components, tDNA and the virulent genes. The both two components which are essential for transformation of plant cells. During the transformation process, tDNA is cut from the Ta plasmid, transferred to plant cells, and integrated into DNA of the plant cell. Scientific classification and taxonomy. Domain Bacteria. Phylum Pseudomonodonta. Class Alpha Proteobacteria. Order Hypomicrobials. Family Rhizobiaceae. Genus Agrobacterium. Species Agrobacterium radiobacter. What is Ta plasmid? Ta plasmid is nothing but extra chromosomal genetic material found in Agrobacterium tumefaciens which causes crown gall disease in certain dicot plant species, for example tomato, potato, asparagus, etc. Also, it plays a crucial role in the ability of Agrobacterium to transfer genetic material into plants, which can lead to the formation of crown gall or tumor on the plants. Ta plasmid was developed in 1980s from various observations. Ta plasmid can be transferred to plant chromosomes, and also it has the ability to transfer other foreign DNA sequences into plant chromosomes. Ta plasmid can lead to changes in the cell growth and differentiation. Morphology. The cells of Agrobacterium are typically rod-shaped or bacillus, and it has elongated and cylindrical. The cell wall has a double membrane structure which contains peptidoglycan, lipopolysaccharides, and other proteins. The cells are motile due to the presence of flagella. Flagella serves various functions, including locomotion, propulsion, and sensory perception, often used by microorganisms to move through liquid environments such as water. The bacterium also possesses pillae or thin-like hair-like appendages on the cell surface. It is also play a crucial role in the attachment of Agrobacterium tumefaciens to plant host cells during the infection process. They generally grow in the range of 0.5 to 1 micrometers in width and 1 to 3 micrometers in length. The cells appear as colorless or pale like non-pigmented cells. Agrobacterium tumefaciens is a facultative anaerobe, which means that it can grow both in the presence and the absence of oxygen. The colonies of cells on agar plates often appears as small, circular and slightly raised with creamy or white coloration. Mode of transmission. Agrobacterium-mediated transformation. First, the tDNA plasmid was removed from the bacterium, and then the plasmid was cut by using the restriction enzymes. and then the foreign DNA was cut with the same restriction enzymes and inserted into plasmid. The tDNA carrying the gene and incorporated into plant cells chromosomes. And then the plant cells were cultured. A transgenic plant is expressing inserted gene is generated. Signal recognition by Agrobacterium tumefaciens. The bacteria infects only wounded tissues of plants and doesn't infect the healthy plants. The wounded tissue or site induces the production of phenolic compounds such as acetoserongone. So acetoserongone is nothing but a natural phenol is composed that plays a significant role in microbe interactions, particularly in the process of genetic transformation or gene transfer in plants. The cells are attracted towards one by chemotaxis. Chemotaxis is a process by which microorganisms like bacteria move in response to chemical gradients in the environment. The lipopolysaccharide aids in the Agrobacterium tumefaciens to the plant. tDNA processing. The virulent genes mediates the processing of tDNA transfer, excision and integration of tDNA into nuclear genome of plants are accomplished by group of proteins or encoded by their genes. As I already said, the acetoserongone is recognized by Vir-A, which causes autophosphorylation. Autophosphorylation is nothing but process in which the enzyme phosphorylates itself by adding phosphate group to one of its own amino acid residues. It is a key regulator mechanism in cell signaling and also plays a crucial role in various cellular function. The autophosphorylated form of virulence A phosphorylates virulence G, activating the virulence gene and causing it to express tDNA is created as a single strand after virulence gene activation. The 25 base pair border region is recognized by two virulence D proteins, virulence DN1, which causes cleavage on both right and left borders of the both strands of tDNA. In order to ensure that 5' end is the 5' end in later stages, virulence D1 is then removed from T strand, while virulence D2 is tightly bonded on the fifth end. tDNA traveling into plant host cell. The plant cell is where the single-stranded tDNA leaves the bacterial cell membrane and enters to Ti plus. The single-stranded nucleic acid binding protein called virulence E2, which is produced by virulence E genes, build up in bacterial cytoplasm and enters the plant cells by endocytosis. Endocytosis is nothing but a cellular process by which cells engulf and internalize particles, molecules, or other substances from their external environment. It plays a crucial role in nutrient uptake, cell signaling process, and removal of waste materials. The virulence gene attaches to single-stranded tDNA in the cytoplasm of plant cells to stop endonucleases from degrading it before it enters the plant nucleus. The T-strand virulence genes get together, they make up the T complex. The plant Ti complex, endoplasmic reticulum network in the plant cytoplasm, transports cytoplasm to the nucleus, and the virulence protein aids in this movement. Integration of tDNA in the plant genome. Nuclear binding proteins like virulence D2, virulence E2, and virulence F unbinds from tDNA once within the nucleus. Actually, it's still unclear how tDNA integrates into host plant genome at the molecular level. tDNA integration happens randomly throughout the plant genome, not just in hypermethylated areas or transcriptionally active. So, expression of tDNA in the plant cells. Phytohormones accumulate when the auxin and cytokinin coding genes are expressed in tDNA. Auxin and cytokinins are nothing but plant growth hormones that plays an essential role in regulating various aspects of plant growth and development, like auxins, cell elongation, apical dominance, root growth, tropism, as well as cytokinins, cell division, delay in senescence, apical dominance, and counteraction, nutrient mobilization. These two hormones often work in conjunction to regulate the plant growth and development. Auxin and cytokinin can interact to control the formation of root and shoots during tissue culture or regeneration experiments. The balance and interaction between these hormones are crucial for maintaining proper plant growth, structure, and response to the environment. Phytohormone ratio causes uncontrolled plant cells proliferation, which results in formation of tumors. So, this is the disease cycle of Agrobacterium tumefaciens. So, the bacterium first attaches to plant cells and enters, and then multiply, and then the tDNA leaves bacterium and enters the plant cells and integrates itself into plant chromosome, causing the transformation, and then they divide rapidly. They leads to gall formation, which exerts a lot of pressure on surrounding tissue. The tumors are easily induced attached by surrounding invaders. Bacteria from gall surface move in soil due to tumor breakage. Plants infected by ground gall, and then the steps involved in tDNA transfer. So there are 7 steps are involved in tDNA transfer. Signal recognition by agrobacterium, attachment of agrobacterium to plant cells, virulent gene induction, tDNA strand production, transfer of tDNA from bacteria to plant cells, integration of tDNA into plant genome, expression of bacterial cells and synthesis of proteins. Symptoms. Symptoms are like rough, abnormal galls on the roots, abnormal growths 2 inches or larger in diameter that appears on lower branches and the stem. The plants with several galls may be unable to move water and nutrients up the trunk and become weakened, stunned, and unproductive. Young plants can be killed by developing gall tissue. They can grow mostly during the warm months of the year. Features. They are responsible for crown gall disease. They are more than 200 kilobase pairs, contains tDNA, up to 15 to 30 kilobase pairs, integrates into plant chromosomal DNA. tDNA region has 8 genes like auxin, cytokinin, opine catabolism, origin, virulent gene, etc. Applications. TA plasmid is the most widely used vector which aids in the development of modified plants with the help of recombinant DNA technology. In genetic engineering, it is used to introduce foreign genes into desired plants. The tumor-inducing and opine synthesis genes are removed and replaced with the gene of interest. The resultant plant is called transgenic plants. Transgenic plants have their genetic material modified by introduction of one or more genes from another organism. The introduced genes are typically selected for specific traits or characters that are designed in transgenic plants. Treatment. Crown gall disease is caused by Agrobacterium tumefaciens, as we already know, and the bacteria does not respond to antibiotics in the same way that many pathogens do in humans and animals. But Agrobacterium radiobacter K84 is an antibiotic that have been developed for biological count of crown gall disease in certain plants to control. They are rather used as a biocontrol agent. Factors influencing TA plasmid. There are numerous methods that are employed to obtain transgenic plants, some of which includes agrobacterium-mediated transformation, particle bombardment, electroporation, protoplasters mediated by PEG, polyethylene glycol, and liposome-mediated transformation. Among all these methods, Agrobacterium results in single-copy transgenes, which are comparatively stable than multiple gene copies. The process includes cell density, plant species, plant growth regulators, and environmental factors. In order to develop an efficient transformation protocol, it's necessary to find the right combination of these factors, like explant, selecting the explant wounding, plant species and genotype, antibiotics, plant growth regulators, light, temperature, and agrobacterium strains. Disadvantages. It's very large and low copy number in agrobacterium, difficult to isolate and manipulate in vitro, that is lab techniques. do not replicate in E. coli, the efficient host for genetic manipulation. tDNA regions from wild-type TA plasmids are generally large, do not contain unique restriction endonuclease sites suitable for cloning gene of interest. So, agrobacterium-mediated gene transfer. It is a widely used method in biotechnology for introducing foreign genes into genomes of plants. The tDNA segment becomes integrated into plant genome at specific location. This integration is semi-random but often accompanied by disruption of gene in the plant genome. Agrobacterium-mediated gene transfer is a precise and efficient method for creating transgenic plants and has been instrumental in crop improvement, biotechnology research, and development of genetically modified crops. It allows scientists to introduce specific genes of interest into plant genome, potentially conferring traits like resistance to pests, herbicides, or environmental stress, as well as enhancing nutritional content. So let's discuss about the mechanism. The mechanisms involve sensing of plant chemical signals, and inducing of virulence proteins. So the cells are first released by the wounded plants by perceived by virulence A and virulence G, two component systems of Agrobacterium tumefaciens, which leads to transcription of virulent genes promoters and expression of virulence proteins. tDNA processing. tDNA is nicked by virulence D2 from the T region of TA plasmid and forms a single standard linear T strand with one molecule covalently attached to 5' end of the T strand. Attaching of tumefaciens to plant and transferring of T complex to plant cells. The cells attach to plants and transfer the T complex from the plant cells by virulence transport system. Targeting of T complex to plant cell nucleus and integrating of tDNA into plant genome. So the T complex is transported into nucleoplasm under the assistance of some host proteins and then integrated into plant genomic DNA. Expressing of tDNA in plant cells, inducing of plant tumor. The genes encoded phytohormone synthesis that leads to uncontrolled cell proliferation of plant cells and opine synthase that provide nutrient compounds to infecting bacteria. So thank you.